YouTube channel. So here in this video I will be discussing about the mechanism of infection of the coronavirus in the human body in a molecular way. So we are very much worried about the coronavirus, so let's know that how it is infecting our body, what is the mechanism of action. Okay, so let's start this story with these two funky guys, okay, that are over here. Both of them are infected with coronavirus. Okay, so let's say that this is the virus and both of them are infected with this virus, right? But they are having different features. For example, this person is not showing any sign and symptoms, but this person can have the capability to spread the disease. Okay, and this person is having infection along with that. This person is showing the signs and symptom, right? So both of them are infected with virus and they are capable of spreading the disease or spreading the virus to other person, normal person, that is over here. Okay, so from this two person, this normal person is getting infected. Okay, so after getting infection, as you all know that this is the virus which infects our upper respiratory tract, right? So this virus will take entry in this normal person through by these two individuals. Then that virus will infect the upper respiratory tract of that particular person, this person. Now as the virus has entered in this fellow, now what the virus will do, the virus will move. Now it is infected and the virus will move to lungs, and in the lungs, you know that alveoli are there. Okay, and I am just zooming in, I am just doing one alveoli in a bigger way. So this is the alveolus. Okay, and this alveolus is consisting of two type of cells. One is the type 1 pneumocyte cell, another one is the type 2 pneumocyte cell. I have marked two type of cells in different colors. As we have seen here, the black dots or black circles are the type 1 pneumocytes. This is type 1 pneumocyte, and this red cell is the type 2 pneumocyte cell. Okay, both are having different functionalities. For example, this type 1 pneumocyte cell is helping the lungs for gaseous exchange, right? And this type 2 pneumocyte cells are generating this type 1 pneumocyte cells, as well as this cell is this cell produces the surfactant. Okay, that is very much important for the surface tension, for the contraction and repulsion of the alveolar cells. Okay, so let's see what this virus is doing with these two type of cells. So if we further look this particular circle region in a zoom configuration, then we can see such kind of bigger picture. Okay, so this particular red cell is nothing but the type 2 pneumocyte cell, and these cells are type 1 pneumocyte cells, right? And as I have already shown that the structure of the virus, this is the virus which is present inside this person and infecting these lungs, and inside the lungs it is attacking this alveoli. Okay, alveolus. Now the virus will infect this type 2 pneumocyte cell with its spike protein, and a particular receptor is present on this type 2 pneumocyte cell, that is known as ACE2 receptor. So this is the receptor known as ACE2 receptor, that is angiotensin converting enzyme 2 receptor. Okay, and as soon as it binds with this receptor, it will be engulfed inside the cell, or it will be endocytosized inside the cell, inside this pneumocyte cell, type 2 pneumocyte cell. Now inside the cell, inside this type 2 pneumocyte cell, such kind of endosome will be formed, along with inside which the virus is present, right, as you have seen over here. And as it is entered, this particular endosome is formed inside this type 2 pneumocyte cell. It will be fused with the lysosome, And what it will do, the lysosome, what it will do, it will make the environment of this endosome acidic. So in this acidic environment, the virus will release its RNA. Okay, so that is the concept behind which the hydroxychloroquine is working, because hydroxychloroquine will make this environment of this endosome little bit alkaline. It will actually increase the pH. As a result of that, the RNA cannot be come out. Okay, so that is another issue. I will make a separate video on that particular matter. So let's concentrate in this particular region. So here the endosome is there, and inside the endosome the virus is present. Now, as soon as it is fused with the lysosome, it will make this environment of this endosome little bit more acidic, and the virus will release its genetic content, that is the positive single stranded RNA, sense RNA. Now, once after the releasing of the RNA in the cell, host cell, that is the type 2 pneumocyte cell, this is the RNA of the virus. Okay. Now, as the RNA is released inside the cell, now it will go and bind with ribosomes that are sitting on the endoplasmic reticulum and synthesize the proteins, different type of proteins. Those proteins are known as the polyprotein. Okay, so let me mark those proteins with black. So these are the polyprotein. Let me tell you one thing, that as you know that the endocytosis is mediated by basically the clathrin protein. Okay, but in case of the coronavirus, it can enter the cell, or it can be endocytosized inside the cell without having the activity of clathrin coated protein, right? So this is another thing. Now, once the RNA is translated, the proteins are produced, the polyproteins are produced. Now this protein will be trimmed with another enzyme known as the proteinase enzyme. Okay, so these proteins are trimmed with another enzyme known as proteinase. This proteinase will trim these polyproteins into different functional proteins that will make different parts of its body. Okay, like S protein and N protein, or nucleocapsid protein, then membrane protein, like that. Now, after the formation of the protein, it will be followed the track through ERGL system. Okay, and from the Golgi body, the virus will be released. The virus will be formed. Let's say this is the vesicle that is produced from the Golgi bodies, and inside the Golgi body, the virus is present. This is the virus. Now, what will be happened? This is one function. At the same time, another function is also going on, that is, this RNA will be taken by another enzyme, that is RNA dependent RNA polymerase enzyme. RNA dependent RNA polymerase enzyme. So this is the RNA polymerase enzyme, and this enzyme will multiply or produce a multiple number of mRNAs of the same copy. These are nothing but the viral RNA. Okay, the RNA of the coronavirus. So both of this protein and RNA will be assembled, and they will make the complete functional virus. Then it will be it will go to outside by the exocytosis. Now here the interesting thing is that at the time of trimming those polyprotein, it release some unused protein. Okay, so those unused proteins, so let's say at the time of trimming such red dots are produced. So these red dots are nothing but the unused protein that will not be used for the formation of the body of the virus. Okay, so now this is known as the inflammatory mediators. Inflammatory mediators, I am writing in the short form. Okay, inflammatory mediators. Now what this mediator will do, as these mediators are released at the time of this trimming of this polyprotein, it will trigger the macrophage. Okay, so this is the macrophage, and this mediator will bind on the surface receptor of the macrophage. So this is the macrophage. Okay, and as it binds with this particular trimmed polyproteins, then after binding it produces a different type of chemical secretions. Okay, that are nothing but the vasodilator. So for example, we can say that it will produce soon after the binding of this protein, this inflammatory mediator, the macrophage will produce interleukin 2, 7, 6, 10, IP10, then GCSF, that is granulocyte couple stimulating factors. Okay, and many others. Another is that tumor necrosis factor alpha. Okay, so these are secreted by the macrophage once it is bound with the polyprotein, sorry, the trimmed part of the polyprotein. Now once these chemicals are secreted in the bloodstream, it will go and affect on the blood vessels, right? It will go and affect on the blood vessels. So as a result of that, the endothelial cells of the blood vessel become constricted. Okay, it will start constriction. Now once the endothelial cells are constricted or condensed, now it will create the gap between the cells, right? As a result of that, the permeability will be increased in that case, and from this blood vessel the plasma will rush into this particular space. So here the plasma will be accumulated. So these orange color things are the plasma, and also this plasma will force this alveoli on the upper side. As a result of that, what will be happened, and previously as the virus is infecting the type 2 pneumocyte cell, that means it the surface tension will be increased. So as the surface tension of this particular situation is getting increased, so it will cause the higher collapsing pressure. So in that case, as the higher collapsing pressure is there, so it will actually shrink. It will actually make it in a very smaller, in a very constricted way. Okay, so it will make this alveoli in a very smaller in size. So what it will do, it will create a pressure. So let me make it with a different color. So this plasma will create a pressure on the surface of the alveoli. As a result of that, the size will be reduced. Okay, the diameter will be reduced, and also some plasma will try to take entry inside the alveolus. Okay, and along with that plasma, some neutrophils will also enter, and you know the function of the neutrophil, it will create the reactive oxygen species, and that will kill the type 1 pneumocyte cells rather than killing the viral particles, right? So in that case, once the type 1 pneumocyte cells are getting infected or getting attacked by this neutrophil, then what will be happen? The gaseous exchange will not be occurred. Okay, and after that, what we can see here, we can see the accumulation of the plasma inside the alveolar space, and along with the tissue debris, that may be the type 1 pneumocyte or that can be the type 2 pneumocyte cells. Okay, so that will be accumulated to form the consolidation. Okay, as a result of that, the space will be filled. Okay, now the space will be reduced in this case. So as a result of that, the person having the COVID-19 disease will feel the breathing problem. Okay, now this is one scenario. Another scenario is that once these chemicals are secreted from the macrophage, it will go and trigger the hypothalamus, and in the hypothalamus, once it triggers the hypothalamus, then it will release, hypothalamus will secrete the prostaglandin 2 that will increase the temperature of the body, right? Now this is one kind of phenomenon that is going on, and here these chemicals these chemicals are getting secreted right after the entry of this virus inside the cell. Okay, now another secretion is also there, that is started at the time of attachment. Okay, so let's say this is the type 2 pneumocyte cell and this is the ACE2 receptor. ACE2 receptor. And this is the virus. Once this virus, the spike protein, is attached with this ACE2 receptor, then it will undergo some this ACE2 receptor in the intracellular domain will trigger some phosphorylation events. Okay, so it will activate soon after the binding of this virus with the ACE2 receptor of the type 2 pneumocyte cell, it activates casein kinase 2 protein. Now this casein kinase 2 protein will phosphorylate the serine amino acid of the ACE2 receptor that is present in the cytosolic domain. Now once this serine is phosphorylated by the casein kinase 2, it will further activates by the phosphorylation events another protein, that is RAS. Then it will further go in the downstream process pathway cascade, that is RAF, then make, then ERK 1 or 2. Now this ERK 1 and 2 will activate another transcription factor, that is AP1, and this AP1 will go inside the nucleus and bind, it will bind on the DNA and transcribe a particular segment of the DNA. So that is the RNA transcribed from this part of the DNA, and this mRNA is called CCL2, that is chemokine ligand 2 or CC motif ligand 2 protein. Okay, and that particular that RNA will go to the cytoplasm, and in the cytoplasm this RNA will produce the CCL2 protein. This is CCL2 protein. Now this CCL2 protein will go to the outside and it causes the lungs fibrosis. Okay, so this is the very short and brief video, that is not much more details. So this is all about the COVID-19, the mechanism of the coronavirus in the human body. And if you like this video, please share and subscribe my channel. Okay, so thank you.